Hi friends, so let's continue with our biochemistry lectures. We are discussing glycolysis, and in glycolysis we have discussed some two irreversible steps, main these two steps in which glucose is converted into glucose 6-phosphate, glucose 6-phosphate is converted into fructose 6-phosphate, and this fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Now after this there is a very tragic event which happens. Tragic event for our 6 carbon molecules, because now they will break up. Okay, the 6 carbon molecule which we have right now, which is 1, 2, 3, 4, 5, 6 carbon molecule, which is fructose 1,6-bisphosphate, and it has phosphate at carbon number 1 and carbon number 6. It will break up. Okay, now let me tell you what do I mean by that. So they will break up into 2 pieces. Okay, if you take 6 carbon molecule and you break that into 2 equal pieces, how many carbon molecule you will get in one piece? Of course you will get 3 carbon molecules. So you have 3 carbon molecule over here and 3 carbon molecule over here. Okay, here at first position you have our, yes, this is phosphate group, and at here it was your phosphate group. Here it was first, second, third, fourth, fifth, sixth carbon. Now here it will be first, second, third, and first, second, third. So you have 2, 3 carbon molecules over here. This is irreversible step. Okay, and the enzyme which does this, the enzyme is called aldolase. The enzyme which does this is called aldolase. Now you have to understand that what are these molecules. Let me tell you first. Fructose 1,6-bisphosphate will be divided into 2, and this molecule is dihydroxyacetone phosphate, DHAP. Okay, and second molecule which you will get is, it is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Okay, generally this DHAP doesn't have any active metabolic role in any pathway, so this DHAP or dihydroxyacetone phosphate will be converted back to glyceraldehyde 3-phosphate. Okay, and the enzyme which does this is triose phosphate isomerase. Here the enzyme is triose phosphate isomerase. Triose phosphate isomerase. Okay. So see what has happened. Let us see a little bit in more detail here. Here we were having our nice fructose 1,6-bisphosphate. See, we have talked about fructose 6-phosphate ka structure, correct? In that we have attached the phosphate position as 6. In this structure you just have to add phosphate position at 1. So let's see. So this is 6 carbon molecule. Okay, your fructose is 6 carbon molecule, and at first and this is your first carbon and phosphate at your 6th position, correct? And here you have OH group, correct? And here you have O, here you have H, and this is your OH, H, OH. Oops, sorry, I think double bond loop, of course. Yes, so this was our fructose 1,6-bisphosphate. Now what aldolase does? Now aldolase is your, what is aldolase? Aldolase is your reversible enzyme. So you have this three carbon molecule which is This will remain a ketonic group. Okay. So this is your, and you will get glyceraldehyde 3-phosphate. Now this is and glyceraldehyde. Okay. Orientation गलत हो गया, sorry. Yes. And this is your glyceraldehyde 3-phosphate. And this enzyme is aldolase. This aldolase is very interesting because you have aldolase of two types, aldolase class 1 and aldolase class 2. Class 1 and class 2. Okay, this class 1, specifically this class 1 is used, or you can see this class 1 in animals and plants. And if you talk about class 2, this class 2 you can see in bacteria, fungus or fungi, or some algae. Okay, so this class 1 is very different, class 2 is different. They do the same role of converting fructose 1,6-bisphosphate into your 2-triose molecule, that is DHAP and G3B. Now why I am telling you this? Because there are, see, if there are some drugs which are targeted to specific bacteria. Okay, if you can target drug on class 2-1, suppose we are infected with some bacteria and we target exactly this aldolase class 2, so that aldolase class 2 will not interfere with our aldolase. Okay, so that's how this drugs, some drugs are designed on this principle. Okay. So what do we have now? We have 2 molecules of glyceraldehyde 3-phosphate. Okay. Now we have 2 molecules of glyceraldehyde 3-phosphate. We started with glucose. We have invested, right? How many ATPs we have invested here? We have invested 2 ATPs here and at this stage, here and here. Okay. So we have invested this energy and now we are getting 2 molecules of 3 carbons. So we have split the fructose. Now let's see what happens. Now I will talk about only glyceraldehyde 3-phosphate. That is true for even this glyceraldehyde 2-phosphate, 3-phosphate. Okay. So let's see what happens to this glyceraldehyde 3-phosphate. Okay. Now this glyceraldehyde 3-phosphate will enter into a very interesting phase. Okay. So this glyceraldehyde 3-phosphate, yes. If we talk about so of course we are talking about 2 molecules of glyceraldehyde 2-phosphate, glyceraldehyde 3-phosphate. So in this step, in glyceraldehyde 3-phosphate, you can understand what will happen, because the next step is, okay, the next step is 1, 3. So of course if I am talking 2 number, that means there is phosphate coming somewhere. Okay. So next we have 1, 3 bis phospho glycerate. This is 1,3 bis phosphoglycerate. Now 1,3 bis phosphoglycerate, it tells us, see, glyceraldehyde 2-phosphate into 2, let's write here into 2 because we have 2 molecules. So of course we will get 2 molecules of 1,3 bis phosphoglycerate. So 1,3 means there is one more phosphate which is coming at position number 1, because 3 position के ऊपर यहाँ पे there was already a phosphate group. This was your glyceraldehyde 3-phosphate. And now what has happened? Now your molecule is, yes, now your molecule is 3 carbon and it has 2 phosphate. Now you will ask, sir, then are we investing one more ATP over here? The answer is no. Okay, the answer is no. So you will ask from where this phosphate group is coming. The phosphate group is coming, okay, the phosphate group is coming here with an inorganic phosphate. Okay, this enzyme which we are talking about, this enzyme, okay, let us talk about this name first. This enzyme is glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. Now, when we talk about dehydrogenase, that means something is related with hydrogen. There is dehydrogenation reaction. We have studied in enzymes what is dehydrogenation reaction. Correct? When we talk about glyceraldehyde 3-phosphate dehydrogenase, it does two things. Okay, it does two things. This is kind of an oxidation step. Let me tell you. Okay, this does two things. This glyceraldehyde molecule, this molecule can take the, let's say this is the enzyme. This is the enzyme. Glyceraldehyde 3-phosphate dehydrogenase G3PDH. Okay, this will take NAD and will convert into NADH. Yes, what we are doing, let me write in red, so there is something energy, an important step. This is NAD and NADH. Here we are taking NAD, okay, and it is giving NADH. And this enzyme is also taking inorganic phosphate, okay, and attaching this inorganic phosphate here at first position. Okay, taking inorganic phosphate and it is attaching at the first position. So this is 1,3, this is 1,3 bis-phosphoglycerate, and we have two molecules. So how many NADH we are getting? We are getting 2 NADH. Okay, so whatever we are talking about here, we are talking about two things. 2 glyceraldehyde 3-phosphate molecule. So how many phosphate will be involved here? 2 phosphate, because we have 2 phosphorylate 2 molecules. NADH, we will get NADH from 2 molecules. So how many NADH we have now? We have 2 NADH. So we are starting getting our money back. Now the first money which we have got is not really money. Okay, this money is kind of a this money is like a check, bank check. Okay, now this bank check, if you have in your hand, it's useless. Correct? For that, the bank should be open and the bank employees should be there. Only then you can take this check into the bank and then you can cash with some money. Exactly like that. If we're talking about glycolysis, okay, if you're talking about glycolysis, glycolysis will move to pyruvate, pyruvate will go into Krebs cycle, and there will be generation of NADH, no doubt about it. And this NADH will go into a process which is called electron transport chain, and this is the bank. Okay, this bank is your mitochondria. So you need mitochondria and the enzymes which runs this electron transport chain. We need oxygen for that. So if you have oxygen and you have electron transport chain, then you can catch this NADH. So what is it worth? ये कितना रुपए का है NADH? So NADH is equal to, so 1 NADH is equal to 2.5 ATP. 1 NADH is equal to 2.5 ATP. So we have 2 NADH. So it's equal to 5 ATP. So we have kind of got we are getting our money back. Kind of, we are hoping at least that we have invested 2, now we have got 5, presumably 5 ATP from glycolysis yet. But let's talk about only 2 NADH. ATP की हम लोग बात करेंगे जब when we talk about electron transport chain, and we'll see the situation. Okay, this is your 1,3 bis phosphoglycerate. Okay, Now this 1,3-bisphosphoglycerate. Now why I'm talking about this is a very important, or not very important, but actually an oxidation reaction. Let me tell you. So we have talked about, this is your, yes, this was our 3-phosphoglycerate, correct? Glyceraldehyde 3-phosphate. Okay. Now this glyceraldehyde 3-phosphate, you will see this glyceraldehyde 3-phosphate. This is your aldehyde group. Correct? This is your aldehyde group. Now this aldehyde group will be converted into an acidic group. Now let's see what is our acidic group. We all know what is acidic group over here. Okay. And here there will be one more phosphate attached. And then we have 3 carbon over here, and we have this carbon over attached. ये कार्बन तो है ही, और ये हो जाएगा हमारा OH और H. ये हो गया हम लोग का aldehyde to acidic group. Okay, so we have converted the aldehyde to the acidic group. Now we have, yes, 1,3. This is nothing but your 1,3-bisphosphoglycerate. Okay, 1,3-BPG. And this 1,3-BPG, I will tell you something very interesting about this. Let me get into it first. Okay, this 1,3-BPG, which is 1,3-bisphosphoglycerate, this enzyme also is very important, glyceraldehyde phosphate dehydrogenase. So the next step which we are talking about, which will take 1,3-bisphosphoglycerate and it will convert into 3-phosphoglycerate. That means if we talk about this step, let's say, okay, 1,3-bisphosphoglycerate is converted into, yes, it is converted into 3-phospho 3-phosphoglycerate. Okay, and the enzyme here is, so you can see in the picture itself, or in the name itself, what has happened. 3 molecules, and there is just phosphate at one position. So what do you mean by this? That means we have taken out one phosphate from here. So if we have taken out one phosphate from here, so this molecule is 3-phosphoglycerate, and the enzyme which work here is phosphoglycerate kinase. The enzyme here is phosphoglycerate kinase. You will ask, kinase क्यों सर? phosphate तो ले रहा है, दे नहीं रहा है. Kinase इसलिए because ये phosphate दे रहा है, it is giving phosphate to ADP. Okay, so now it also, so let's say this is the enzyme. Okay, phosphoglycerate kinase, and it is taking, yes, it is taking ADP and giving ADP, and of course this is 2 बार. So we have generated 2 ATPs over here, correct? Because we are talking about one molecule, but we have got 2 molecules of glyceraldehyde 3-phosphate. So of course 2 molecules of 1,3-bisphosphoglycerate and 2 molecules of 3-phosphoglycerate. That means 2 molecules से phosphate निकाला जाएगा और 2 ADP को ATP में convert किया जाएगा. So now what do we have? Till now we have invested. Investment कितना हुआ अभी तक? Investment is 2 ATP, and what we are getting in return कितना मिला है? Just now we have got 2 ATP, and of course we have also got 2 NADH, but we will just talk about ATP for now. So we have got 2 ATP. That means जितना invest किया था उतना return आ गया in this step. Okay. And if you talk about, see, this is the kinase, and this is the only kinase in whole glycolysis pathway which is reversible, because the delta G in this is not very less. Okay, that's why it depends on the concentration of 1,3-phosphoglycerate and 3-phosphoglycerate. Whoever its concentration is more, it can move like this. So only this kinase in whole glycolysis pathway is reversible. Otherwise, every kinase in glycolysis pathway which we have talked about till now, which is hexokinase and phosphofructokinase, and there is one more kinase which will be coming, which is pyruvate kinase. These all three kinases are irreversible, but only this kinase, which is phosphoglycerate kinase, is reversible. Okay, yes. So I will take you little bit sidetrack from here because it's very important. Okay, you have heard about arsenate poisoning. Okay, when a person takes arsenate, okay, this arsenate is very problematic compound because, okay, because this arsenate mimics, okay, this arsenate and your inorganic phosphate, okay, they have a very similar molecular compatibility. So wherever this inorganic phosphate is going and attaching, okay, like there is some enzyme which is taking this inorganic phosphate and doing its job, okay, and attaching it to the molecule. So wherever this kind of stuff is happening, that those reactions, that pockets or that active site, even arsenate can bind. Okay, and if arsenate binds, there will be lot of problem, specifically in this cycle. Correct. So this so what I want to say is this arsenate and phosphate have very similar molecular compatibility. So let's see what happens if you talk about an arsenate over here. Okay, if somehow arsenate is binding in here. Now if you talk about arsenate over here, okay, let's say this glyceraldehyde 3-phosphate dehydrogenase enzyme, this do not add a phosphate group over here, but that enzyme adds an arsenate group. Okay, that enzyme adds an arsenate group. Let's say this is an arsenate group. This is an arsenate group. इस arsenate group गलती से हो गया, glyceraldehyde 3-phosphate को लगा that it is an phosphate, but it has attached arsenate. Now this arsenate is with very loose compatibility and everything is that. So in next reaction, when 1,3-bis phosphoglycerate will be converted into 3-phosphoglycerate, it will convert. No doubt it will convert, but you will not get conversion of ADP to ATP in those cells. Are you understanding? Okay, let me make a point over here. So you are having glyceraldehyde 3-phosphate. Okay, there was molecule glyceraldehyde phosphate dehydrogenase. Okay, and then you got 1,3-bis phosphoglycerate. Okay, and then you got 3-phosphoglycerate. Now, if, and here the enzyme is attaching, is taking ATP and converting into ADP. Okay, no, sorry, I'm very sorry. I'm very sorry here doesn't happen like that. So this is how you make mistakes. So you have here enzyme glyceraldehyde 3-phosphate dehydrogenase. So this enzyme takes inorganic phosphate. Suppose it doesn't take inorganic phosphate and it takes arsenate. So your 1,3-BPG will be there, no doubt. Okay, but at position of phosphate, arsenate will join. And when this will be converted into 3-phosphoglycerate, okay, in this 3-phosphoglycerate molecule, here where ATP was converted into ADP was converted into ATP, यहाँ पे जो conversion हो रहा था ADP का ATP से, ये नहीं हो पाएगा, because phosphate है ही नहीं, वहाँ पे arsenate है, and arsenate का कुछ नहीं हो सकता with ADP. So in this condition, what happens? In this condition, there is a problem that your cell which we are having, where was that? Okay, I have rubbed there, I guess. So here what is the problem? Here we are investing 2 ATP till now, and we were getting back 2 ATP, but this 2 ATP will not be there because we have not able to convert this ATP to ATP. Okay, ये 2 ATP जो मिल रहा था हम लोगों को वो नहीं मिलेगा, so of course the cell will be energy deficient. Okay, so this thing we have to, and there are some couple of more reactions in future you will see in different pathways where inorganic phosphate is binded, and their arsenate also interferes and it causes lots of problem. So that's why this arsenate we are talking about. Okay, then apart from this arsenate, I will also like to tell you there is one more slight outtract pathway. Okay, there is slightly outtract pathway. Let me draw over here. There is no space in this board is small. So yeah. Let me run this. So there is one more pathway over here which is called Rapoport-Lewis pathway. Rapoport, let me tell you the spelling. Rapoport Lewis pathway. What happens in this Rapoport-Lewis pathway? Let me tell you. There are some RBCs. Some let's say 20-30% RBCs which runs this pathway. Which pathway? Let me tell you. Okay. Sometimes what happens? You are glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate. This is your glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate is converted into 1,3-bis-phosphoglycerate. Correct? Glyceraldehyde 3-phosphate is converted into 1,3-bis-phosphoglycerate. And in this reaction which happens in RBC, no doubt, okay, but in some RBCs, okay, and of course the enzyme here is, what is the enzyme? Glyceraldehyde phosphate dehydrogenase. This is the enzyme which does this reaction. Okay, but sometimes what happens, this take a little bit sidetrack. Now what is the sidetrack? That there is an enzyme which is called glyceraldehyde 3-phosphate mutase. Okay, this is called glyceraldehyde this enzyme is called glyceraldehyde 3-phosphate mutase, and this enzyme makes one molecule which is not 1,3-BPG, but it is 2,3-BPG. What it is? It is 2,3-BPG. Now this 2,3-BPG is very interesting molecule because 2,3-BPG is again a 3-carbon molecule and phosphate at 2 and 3rd position. Okay, so the enzyme here is, yes, okay, sorry, the enzyme here is bis-phosphoglycerate mutase. Yeah, bis-phosphoglycerate mutase. Okay, and this enzyme, what it does, it converts your glyceraldehyde 3-phosphate into 2,3-bis-phosphoglycerate. You are asking why we are stressing more on this? Because this molecule is very important for, sometimes it's very important for RBCs. Now why it is very important? Because this molecule binds to hemoglobin. This molecule binds to hemoglobin. Okay, and when this molecule binds to the hemoglobin, the affinity towards oxygen of hemoglobin will reduce. Okay, now let's say our RBC, see RBC first of all doesn't have DNA, RNA, so the energy requirement in RBC is actually very less. Okay, so if the energy requirement is actually very less, when we talk about this molecule 2,3-BPG, okay, what is this RBC? RBC is like a car which carries oxygen from one place to another place. Hemoglobin is like seats on this car, correct? If the seats are very comfortable and very sticky, okay, so if oxygen comes and binds at hemoglobin, it will not able to get out of that chair. It will not be able to get out of that hemoglobin. Exactly like that, this 2,3-BPG makes this chair little bit uncomfortable for oxygen to sit. Okay, so this oxygen can very easily opt out. So the affinity of hemoglobin towards the oxygen will reduce. Now you ask why we want this? We want this, correct? Suppose, just imagine if there is hemoglobin and if it is not leaving oxygen very easily, and it's going to the place and it's not very easily leaving the oxygen, so there will be a problem. Okay, so to maintain a very good aerobic condition, you need this kind of stuff. Okay, so the affinity towards oxygen of hemoglobin is decreased by this molecule, and that's why this molecule is very important. Okay, yes, and also there is very important, if you talk about, sometimes there are let's say sometimes the tissue needs oxygen. Okay, and if 2,3-BPG is not there, then it can be a problematic, because the oxygen will not be released very efficiently, and the tissue will be in hypoxic for a long time. It's not very good situation altogether. Okay, yes, so this is called Rappaport-Lewis pathway. And this 2,3-bisphosphoglycerate, okay, this 2,3-bisphosphoglycerate can of course escape. What is the reaction after this? 1,3-bisphosphoglycerate के बाद में क्या है? 3-phosphoglycerate. So after this 2,3-bisphosphoglycerate, it can convert into 3-phosphoglycerate. Okay, and here the enzyme is okay. We can talk about this enzyme later because it's not very important enzyme. Okay. So here when we talk about specifically 1,3-bisphosphoglycerate, let's move forward. Got to finish the whole pathway. So this was a slight little outer way for little offshoot of Rappaport-Lewis pathway. This happens in some RBC. Okay, so we were in 3-phosphoglycerate. Okay, now we are talking about 3-phosphoglycerate. So let's talk about 3-phosphoglycerate over here. Okay. Here we are talking about 3-phosphoglycerate, and this 3-phosphoglycerate is also an 3-carbon molecule. And how many 3-phosphoglycerate we have? We have how how many 3-phosphoglycerate we have? We have 2 3-phosphoglycerate. Here 3-phosphoglycerate. Okay. This 3-phosphoglycerate, how many molecules of 3-phosphoglycerate? Into 2. Okay, ये आपको समझ में आ रहा है ना what I am talking about? So this is the molecule of 3-phosphoglycerate. Now this 3-phosphoglycerate will be converted into another molecule which is just a kind of a isomer kind of thing. Okay, and this is again a reversible step. Of course, this is also a reversible step. This is also a reversible step. So now what we are doing, we are shifting the phosphate position. We are shifting the phosphate position at second carbon. Okay. So here it was phosphate position first. So now we just have इसको भर दो पूरा वैलेंसी. ओके, यहाँ पे आ जाएगा आपका फॉस्फेट ग्रुप. यहाँ पे आ जाएगा आपका फॉस्फेट ग्रुप. ओके, एंड दिस here acidic group. Okay, and this molecule is, this was 3-phosphoglycerate, this is 2-phosphoglycerate. Okay, and the enzyme here is phosphoglycerate mutase. Phosphoglycerate mutase, this is reversible enzyme. Okay, it's a reversible enzyme. Again, the delta G is okayish, not very low, so it can jump up at any position. It can go reverse, others forward, depends on the concentration. Yes. Now, some interesting things will happen. Okay. Now, you have this molecule which is 2-phosphoglycerate. Okay, this is the molecule, and we are talking about okay, this is 2-phosphoglycerate. Enzyme just stopped working. This is 2-phosphoglycerate, and this 2-phosphoglycerate molecule is now converted into, okay, this 2-phosphoglycerate molecule. Okay, you remember this was 3-phosphoglycerate. Now we have converted into 2-phosphoglycerate. This is at second position. Okay, after this, now this phosphate position will go a little bit, like little bit loosed. Okay, just ready to opt out. So that's why this molecule becomes very energetic. Okay, so now what is happening over here? The enzyme here is enolase. And what we are doing, we are removing water from this molecule. We are removing H2O. Okay, we are removing, so we want all the energy from it. So we are taking out water now from this molecule. If you take out the water, okay, of course there is no fun to say it's still 3-carbon compound. Okay, इसके बीच में एक 2 बॉन्ड आ जाएगा, बिकॉज यहाँ से निकल गया है एक H, यहाँ से OH निकल गया, यहाँ से OH निकल गया और यहाँ से H निकल गया. Okay, and you still have an acidic group over here. This is double bond, so here will be, sorry, इधर नहीं होगा, इधर होगा O, P, O, phosphate group over here. Yes. Okay, so this is enzyme is enolase. Again, it is reversible step. Okay, and this is the enol bond which has formed, and it is one of the most energy rich bond. Okay, it is one of the most energy rich bond. Of course, the enzyme name is phosphoenol pyruvate. Okay, and this is the most energy rich molecule. Okay, PEP is the most energy. This PEP इसको प्यार से हम लोग क्या बोलते हैं? Phosphoenol pyruvate. Phosphoenol pyruvate is one of the most energy rich molecule in whole biochemistry because it has the most energy rich bond over here, that is enol phosphate. Okay, there are two main energy rich molecule in the whole glycolysis. Let's say one is 1,3-bisphosphoglycerate and other is your phosphoenol pyruvate, and of course phosphoenol pyruvate is one of the most energy rich bonds. Okay, so last, then we are reaching to our end, and of course we have 2 PEP. Okay, so let's see now. Yes, the last reaction what we are having is our phosphoenol pyruvate. Okay, we have phosphoenol pyruvate over here, which is 3 carbon molecule which we have seen, and we have, okay, this is PEP, and this PEP is now converted into a 3 carbon compound without phosphate, that is pyruvate, which was our main goal. Okay, so PEP is converted into pyruvate, and the enzyme over here is, yes, pyruvate kinase, the 4th kinase enzyme in whole glycolysis. And of course there is no fun to say, this is, yes, this is irreversible. This is irreversible. And if you talk about the regulation of this enzyme, okay, we have talked lots about kinase regulation in earlier also. So who are the inhibitor of this enzyme? Okay, ATP is inhibitor. ATP is inhibitor. Okay, then your AMP is positively regulator, and of course there is one more enzyme, one more molecule which we have studied in the past, which is fructose 1,6-bisphosphate. Okay, fructose 1,6-bisphosphate also comes and positively regulate this enzyme. Okay, there is one more regulator over here. There is one more molecule which is here, which is alanine. Alanine inhibits. I will tell you why I am talking about alanine in a minute. Okay, if you talk about phosphoenol pyruvate, let us see the structure once. Matlab you will be very clear what we are talking about. Yes. So this is our phosphoenol pyruvate. Here we have bond, and here we have, and now what we are doing, we are talking about pyruvate over here. This is C, this is 3, 4. And yeah, we are removing phosphate group here. If you remove phosphate group, who is getting the phosphate group? ADP. Correct? ATP. How many ATP is forming here? 2 ATPs are forming. हम लोग ने पहले हिसाब बराबर कर लिया था. Now we have got 2 ATPs extra. Correct? We have got 2 ATPs extra. So this is now, what is this? This is generation of 2 ATP. Okay, we have invested 2 ATP, and we have got, in return we have got 4 ATP. So net profit is of 2 ATP. Of course, we are talking about the other problems like arsenate and all. In that 2 ATPs will not form. The total will be 0 ATP only. Okay, so the pyruvate kinase is again a very well regulated enzyme. Okay, your fructose 1,6 bisphosphate regulates this. Okay, this is called feed forward reaction. Okay, fructose 1,6 bisphosphate in the earlier sections. So that molecule will be coming and activating. Okay, it will start activating this reaction. Okay, so this is all about glycolysis. Okay, so we'll see some of the energetics and everything, some last few touches of glycolysis, and then we will finish glycolysis. Okay, so we'll meet in next lecture. Thank you.